All right. In order to talk about the practical applications of immunology, we first need to dig deeper into that concept of immunological memory, which is what makes your immune system an adaptable defense. So let's take a look at what happens throughout the course of an infection. As you'll recall from chapter 17, the primary response of the immune system happens when antigen is recognized by either the B cells of the humoral branch or the T cells of the cell-mediated branch. In this diagram here, we're concerning ourselves only with the B cells, which are responding to and becoming activated because of their antigen. So on the left-hand side of the screen, you see three different B cells, each of them having three different B cell receptors, only one of which is capable of binding to the particular antigen that's shown here. Because that B cell has recognized the antigen, he will become clonally selected, and as he clonally selects, he will also differentiate into plasma cells, which secrete antibody in high amounts. And very importantly, and something we didn't really talk about in chapter 17 much, was the fact that that activated B cell will also differentiate into memory cells. And the thing about memory cells is that unlike a naive B cell, the memory cell will persist in the body typically for the remainder of your lifetime, and it will constantly secrete small amounts of antibody to that antigen for the rest of your life. So you'll remember the B cell will remain in the lymph nodes, and it will constantly be secreting low levels of IgG into the bloodstream because the immune response is always through the bloodstream. And that's how you can get an idea of your immunological memory by taking a blood sample, because in your blood will be IgG to everything you've ever been exposed to and mounted an immune defense to. So there is memory when you have a T cell response as well. Remember, the T cells differentiate into effector cells, and then those effector cells, after the infection subsides, will become effector memory cells out in the tissue, and then there will also be central memory cells in the lymph nodes, so that you do have T cell mediated memory as well. Just for simplicity's sake, though, in this chapter, we're going to focus on that memory as it is expressed as antibody titer. How that immunological memory looks can be seen on the graph in front of you, where you have antibody titer on the y-axis and time in days on the x-axis, and you're looking at a couple of different things. On the green line is where we're really going to be focusing our attention. That is the total antibody production in response to a particular antigen. The blue line is the IgG antibody that's produced, and the red line is the IgM. Just to keep reiterating the same thing, IgM is typically an indicator of initial infection. IgG is an indicator of a memory to infection. So when you're initially exposed to an antigen, it takes about a week for any antibodies to be measured at all. Remember, that week is the amount of time it takes for the antigen to get to the naive B cell, for the B cell to become activated, and that activation and the production of plasma cells and antibodies takes a while. So you really don't get a good primary response until about 2 weeks after a person has been inoculated with antigen, which again is why those innate defenses we talked about in chapter 16 are so important. Once the acute infection subsides, the overall level of antibody decreases sharply, but as you'll notice from the green line, right after day 21, it never disappears entirely, though the level of IgM will. If you've been exposed to that same antigen sometime later, here's where the beauty of immunological memory comes in. The response is so much faster and so much bigger that would really have made you sick initially will possibly not even make you feel symptomatic at all the second time you're exposed to it. And that's what memory does for you. Memory provides a targeted, smart response very quickly that works really, really well to get rid of all of the invading microorganisms.
Okay, immunological memory involves B cells and T cells, which move via the bloodstream. They're not tending to move within the lymphatic system anymore. They leave the lymph nodes, they go into the bloodstream as a result of being selected and activated by their antigen. So what you see on the left-hand side of the slide here in A is a naive B cell. Remember, naive B cells are those B cells which have not yet seen their antigen. And remember, the B cell can see its antigen and be activated in 3 different ways. It can see its antigen directly, it can receive its antigen from dendritic cells, and it can receive activation from helper T cells if the B cell serves as an antigen presenting cell. Either way, activation occurs, which involves clonal expansion and differentiation into plasma cells, which, remember, are going to secrete IgM. Remember, we talked about IgM in chapter 17 as an indicator of an acute immune response, meaning not memory, but an infection that's being dealt with at the time. But those cells will also clonally expand and differentiate into memory B cells, which are very long-lived, right? Which are not like naive B cells, which have a particular lifespan. These are cells, these memory cells are cells that are going to persist in the body for the remainder of your life. And as we've mentioned earlier, because they persist forever, they have functionally changed your immune system, because now your immune system, in response to that particular antigen specifically, has now changed and has adapted to the presence of that antigen. And that memory is why we call this response an adaptive response, because the memory is what allows the immune system to be functionally different after it encounters antigen. Now, generally speaking, the same thing is true of naive T cells. Naive T cells encounter antigen, of course, always via the presentation of antigen by antigen presenting cells. Remember that naive cytotoxic T cells will receive antigen from all body cells via the MHC class 1 molecule, because their CD8 molecule binds to the MHC class 1, whereas helper T cells, which have a CD4 T cell receptor, will interact with the class 2 MHC molecules of only antigen presenting cells, which we talked about in chapter 17 as being those dendritic cells, macrophages, and B cells that were discussed in detail there. So, regardless of how activation occurred, activation results in memory cells. And again, this memory is what makes the immune system adaptable. So memory B cells reside in the bone marrow, and they constantly secrete a low level of IgG, primarily antibody. And so that's why IgG can be a marker of immunological memory, right? An actual record of every infection you've ever encountered and mounted an immune response to. Effector memory T cells are a type of memory T cell which actually reside in the tissue space. So they hang out in the tissue space and they look for more signs of infection, right? They monitor for a second outbreak. There are also central memory T cells which move through the lymphatic system, and they move through the lymphatic system so that they can be quickly able to activate those memory B cells upon secondary infection. So what we're seeing at the bottom here is antibody and effector T cell responses to a particular antigen, and what we're seeing is 3 different exposures to that same antigen. And it is important to realize that these are the same antigen being encountered. So at the left-hand side, we have the primary or initial infection, at which point the immune system is naive to that antigen. It's never seen it. And so this is Exactly what we saw in chapter 17. We see a very slow immune response, so the body is relying on the innate defenses in order to protect it until about a week has passed and the immune system can come on board, right, with those clonally selected, activated, differentiated plasma and effector cells to fight the infection, and then the infection will be cleared, everything's great, you recover, and your immune system has now learned it's different, it's adapted to that antigen. So the next time you get exposed to that same antigen, you will have a protective immunity, which means the response is so fast and so strong that really you have an inapparent or very mild infection. And that continues throughout the remainder of your life. Notice the x-axis on this graph starts in days between the first two infections, and then notice it moves to years. So you're talking about an exposure to antigen that's occurring more than 2 years after the initial infection. And notice how strong and rapid the response is, right? Which we can see because of the strong, rapid, measurable increase in the antibody and effector T cells. So again, you have such a fast and strong response that really it's either not, you don't even notice you're sick, or it's so mild you wouldn't even really call yourself sick at that time. So immunological memory allows your immune system to learn from its infections and to respond quickly and well to subsequent exposures to those same antigens. And it all relies on the fact that we have those memory cells cruising around the body looking for that antigen. One of the things about those B cells and T cells, which are memory cells, is that they have a higher affinity for their antigen in comparison to their naive counterparts. So we know that they're more abundant because that one cell has clonally expanded into multiple cells, which means that the response of the memory cell is 100 to 1,000 times the response of the primary infection. In addition, the memory affinity, so the affinity of the memory cell to its antigen, is much, much higher. In primary infection, about 1 out of 100,000 naive cells will bind tightly to their antigen, but on secondary infection, about 1 of 1,000 will bind very, very tightly. So you have a much higher affinity of those cells, those memory cells, to their antigen. In addition, the memory B cells do not require the activation of the helper T cell. Remember in chapter 17, we talked about the fact that initial infection, which caused T cell independent B cell proliferation, was very weak, right? Very slow, very weak, very short-lived, and didn't work very well. But B cell activation, which was T cell dependent, was much better, much stronger, had a higher affinity, and overall worked better. Well, it turns out that once you teach the B cell, once it learns, once it clonally expands and activates and becomes this memory B cell, it no longer requires the interaction of the helper T cell in order to respond to antigen, to become activated again. Because all of these memory cells are capable of becoming activated just like the naive cell. So upon activation, they will differentiate, if they're B cells, into more memory cells and plasma cells, which will secrete antibody. And if they are T cells, they will clonally expand and differentiate into both memory cells and effector cells, which are going to go out and fight infection. So memory is better, right? The immune system is better able to fight this infection, both because they're more common, they have a greater affinity for their antigen, and in the case of B cells specifically, all antigens are considered T cell independent, but that T cell independence does not mean that there will be the weak, short-lived, and low affinity response that you see in a primary infection. So that's memory. But this chapter talks about the practical applications of immunology. So what is the practical application of immunological memory, right? Immunological memory simply means that you acquire immunity, your immune system changes via natural means. And what we've talked most about is acquiring immunity via a natural means by way of infection, which we would call an active natural acquired immunity, which you can see there from the dichotomous key. And that's all of what chapter 17 was talking about. You being exposed to antigen, you responding to antigen, and you creating memory to that antigen, which changes your immune system and adapts it specifically to that infectious agent, that antigen. What we really haven't talked much about is the one way that you can acquire immunity naturally in a passive fashion. And what I mean by passive is that you are receiving antibodies. Your body is seeing antibody to infection rather than antigen to infection. And the only way that this is possible naturally is from mother to baby via placental transfer and via breastfeeding. Now, we talked about the fact that IgG, which is a record of all of the infections that you've been exposed to, IgG is the only of those immunoglobulins or antibodies that is capable of being passed through the placenta, and thereby is the only immunoglobulin or antibody that is capable of providing passive, natural, acquired immunity to a baby. Now, that happens primarily via the placenta, right? So if you look at the bulleted points at the bottom, that placental transfer means that when the baby is born, they have a very, very high level of serum Ig antibody, and it's all because their mother passed that antibody to them. But because antibodies don't stick around forever, the serum, right, the blood serum level of antibody in the baby or neonate will start to decline, and it will hit its lowest level at about 3 months of age. Now, it hits its lowest level there, even though there's still going to continue to be a decrease in the amount of maternal IgG over the next couple of months. At 3 months, that's where the baby's immune system starts to kick in and starts to respond to antigen. So the neonatal serum antibody level is lowest at 3 months, and by 5 to 6 months, there are no longer IgG circulating in the baby's bloodstream, and that happens regardless of whether the mother is breastfeeding or not. And the reason for that is because human beings do not have any mechanism to transfer these IgG antibodies from the gastrointestinal tract through the epithelium of the intestine and into the bloodstream of the baby. Now, that is common in other mammals. Mice, for instance, are capable of doing that. But babies, human babies, are not, they have no mechanism for transferring those maternal antibodies into their own blood supply. So at 3 months of age, really, you have a baby who is at their most susceptible to infection because their serum antibody level, right, which is protective, is at its lowest level. Now, thankfully, that baby's immune system comes on board very quickly, and you can see there in the second bulleted point that at 1 year, the IgM and IgG in a baby's blood supply are at about 75% of an adult level, which means that at 1 year old, functionally, a baby's immune system is working at about 75% of maximal. Their IgA, on the other hand, which is one of those antibodies we didn't really talk about, is only at 20%, which leads me to my third and final point, because right now you might be wondering what the benefit, aside from nutritional, breast milk might have for infants, and that benefit is that breastfeeding will provide the baby with high levels of IgA, because IgA is secreted in breast milk, and it is through the breast milk is transferred into the gastrointestinal tract of the baby and is very, very important for fighting mucosal infections of the gastrointestinal tract or enteric tract. And that might not seem very important, right? You might think, well, you're only talking about providing protection against gastrointestinal infection, but gastrointestinal infection is, first of all, very, very serious in young children because it quickly leads to dehydration and malnourishment. And secondly, because of the high incidence of gastrointestinal infection in children. So I don't want anyone to get the idea that what I'm saying is there's no need to breastfeed because we can't transfer antibodies to the blood plasma, because in fact, antibodies will reside in the gastrointestinal tract of the baby and provide much needed protection from gastrointestinal infection there. But again, we haven't talked about any real practical application of immunology there, right? I mean, we have talked practically about why it's important to breastfeed, but now we're going to broaden this scope, right? Use what we know about this natural process to try to create a situation where we can exploit the immune system. So let's take a look at that. We have learned that when you are initially exposed to microorganism, you produce a slow, weak primary immune response, right? Slow because it takes at least a week to come on board, and weak because it doesn't produce a lot of antibodies compared to the amount of antibodies that are produced when you are exposed to that pathogen again. What we're looking at here in this diagram is exploiting the natural phenomenon by inoculating people not with a harmful pathogen, but with an inactivated microorganism or harmless component of a pathogen as what we call an immunization. So an immunization is the inoculation of a person with a weakened or harmless component of a pathogen in order to generate an immune response, in order to teach the immune system about that pathogen. And when you immunize someone, when you inject them with this harmless component, what you're doing is activating the immune system, which is not necessary in terms of primary response because you don't need antibody to something that isn't going to hurt you anyway, but you're also activating memory, and you're very importantly activating memory without causing disease. And in that way, when the person's exposed to the natural pathogen, their immune system is already familiar with it, already knows what to do about it. And so you get that fast, strong secondary response where you get antibody production and T cell response, and you have the response being so quick and so strong that the person doesn't even notice that they've been exposed to that pathogen. So immunizations, which are the catch-all term for all inoculations of harmless microorganisms or components of pathogen, right, which we oftentimes hear called vaccinations, are the first of all of these practical applications of immunology. In fact, they've been around since the 1700s. So this exploitation of the body's ability to learn and adapt to microorganism is the basis of immunizations and vaccinations, which are meant to protect an individual from infection by teaching their immune system something about that microorganism in a safe environment where an infection cannot occur as a response to the exposure. So what we see then is an expansion of acquired immunity. We talked already about natural acquired immunity, where we can actively be exposed to antigen naturally because we get sick, and we mount an immune response. We also have that passive Natural acquired immunity, where mom is transferring antibodies to the baby or the fetus via placental or gastrointestinal tract transfer. And now we see that we have artificial means of acquiring immunity. We can have an actively acquired immunity in an artificial means if you are given an antigen, right, either shot or nose goo or whatever else. And you can receive passive artificial acquired immunity if you are given antibody to something. Now, the thing to remember about the antibodies, right, when you have a passive acquired immunity, whether it's natural or artificial, that protection is going to be short-lived because you've only been given antibody, right? An antibody does not survive in the body forever. On the other hand, whether it's a natural or artificial acquired immunity, if it's active, because you've been exposed to antigen, you yourself are mounting a response which includes the production of memory cells, which means that this will be lifelong immunity.
Before we move into the 21st century and talk about modern advancements that utilize the production of immunological memory, like immunizations, I wanted to just take a few minutes to talk about the history of infectious disease and the idea of an artificially acquired immunity by talking about a disease called smallpox. This is a very old disease. It's one that has been around in the human population for a really long time. In fact, there are Egyptian mummies, including Ramses V here, at even 1500 BC, who showed evidence of smallpox. It was described in China in about 1100 BC, and by 430 BC, there was some idea that people who had survived smallpox could nurse people who were currently ill with smallpox and not contract the disease again. Fast forward about 500 years, in 100 AD, the plague of Antonine killed 7 million people in the Roman Empire. And about 400 years later, the term variola was coined in Switzerland to give this disease a name that could be communicated amongst different cultures, so people could start figuring out that what was called the speckled monster in one place and something else in another place were all actually the same disease, which causes, as you can see from the two photographs here, huge bumps on the skin that erupt into pustules. So the little one here has an active infection, and the black and white photo next to her is a picture of a man whose face has been scarred by those smallpox pustules. So a very disfiguring disease, in addition to the fact that it comes with about a 30% mortality rate. And it was that mortality rate that allowed people to use smallpox in a really terrible way as a biological weapon. There's evidence of the possibility of that being used in lots of different places, most notably the conquistadors bringing smallpox to the New World, we think accidentally, but for sure, once that was understood to be the disease smallpox in the 1600s, the British actually used it as a biological weapon against revolutionary troops in the American Revolution. And in order to try to protect people from infection, this idea that went all the way back to 430 BC, that survivors could nurse the afflicted, was used to protect people in a process called variolation, which we'll talk about in just a moment. But again, to give us some context, just in the 20th century, about 300 million people died worldwide due to smallpox. Variolation is the intentional inoculation of a healthy individual with variola, which you'll remember was coined in Switzerland in the about 500 AD. And the first known incidence of variolation is described in China in about 100 BC, where the crust from erupted pustules, which are drawn here on the right-hand side of your screen, were put into long straws and blown into the nostrils of the person being variolated. So variolation via nasal inhalation happened at about 100 BC, and at about 1715 AD, something called cutaneous variolation was brought to Europe from the East, and that involved taking the scrapings from a smallpox pustule and poking it into the skin of a healthy individual. And the healthy individual, that is very important because you want to catch someone when they're healthy, they're well fed, so that they are strong enough to fight off the minor infection that's going to result because you are inoculating them intentionally with lesions filled with smallpox. In what were the American colonies at the time in 1721, the process of variolation was used to try to curb a Boston epidemic. And the data from that epidemic show that where people had not been variolated, there was about a 14% fatality rate, much lower than what is commonly thought to be the fatality rate of smallpox, which is 30%. But in those that had been variolated, only 2% of people died. And very importantly, for reasons you will see in just a moment, in 1757, a young boy named Edward Jenner was variolated to protect him from smallpox. Edward Jenner was a British physician who worked a lot in rural areas and is said to have overheard a milkmaid say, "I shall never have smallpox, for I have had cowpox. I shall never have an ugly pock-marked face." And he started thinking about this and researching it, and sure enough, it turned out that there was very little data to show that a person who had contracted something called cowpox from working with cows could ever develop smallpox. And the neat thing about that was that cowpox infection in human beings, because they're not the intended host of the virus, produces a relatively mild infection. And it wasn't the first time that someone had thought of doing this, but Jenner was the first person to scientifically go through this process and see if he could recreate what seemed to have been this immunity to smallpox without ever having contracted smallpox in the first place. And the experiment went something like this. There was a milkmaid who had cowpox. Her name was Sarah Nelms, and he scraped some of the pustules from her cowpox, and he inoculated a young boy by the name of James Phipps with the fluid from those cowpox pustules. James developed a mild case of cowpox, and then a few weeks later, James was intentionally inoculated with scabs that were collected from a smallpox patient, and James did not contract smallpox. So Jenner was very excited about this. He published his findings in 1797, and his paper was rejected. But very quickly it caught on because by 1840, the process of variolation, which included the intentional inoculation of a person with smallpox, was banned in favor of what Edward Jenner called vaccination. And I found it really interesting when I was researching this that there was a National Vaccine Institute created by President Jefferson in 1813, far earlier than I had any idea that anybody really thought about vaccines as a public health measure. You might remember from our history of epidemiology that smallpox was the first and at this point only disease that's ever been eradicated globally. That campaign began in 1967 with the same idea that Edward Jenner published in the late 1700s. People were inoculated with cowpox in order to protect them from smallpox, and there has not been a natural case of smallpox anywhere in the world since 1977. So Edward Jenner is kind of known as the father of modern immunization because of this idea that you could use intentional infection with cowpox in order to protect from smallpox. And I mentioned just a few moments ago that it was a relatively mild illness. So you can see a drawing here of an arm that has been variolated, and there's a huge pustule there and multiple smaller pustules all around the arm, and the person would also feel a general malaise and illness, whereas the arm on the right has been vaccinated with vaccinia virus, which causes cowpox, and it causes a much smaller sore there. And Edward Jenner gets kind of a bad rap because it's true, he inoculated a young boy with cowpox to see if he could protect him from cowpox, or excuse me, from smallpox. But in his defense, he did actually leave his house and his estate to James Phipps when he died, and he spent his whole life sitting in his yard at this little hut he called the Temple of Vaccinia, giving away for free vaccinations to anyone that came to his house. And that vaccination was incredibly successful. You see a picture here at the bottom of the screen of two boys. One of them was vaccinated in infancy, the other was not. They're 13 years old, and they were both infected from the same source on the same day. And the boy on the left is in the fully pustular stage, and the boy on the right has only 2 spots, which at the point where one boy is fully infectious, his scabs have already, or his pustules have already scabbed over. So really an amazing advancement in the protection of people from what was a devastating illness. The use of cowpox to protect people against smallpox was an amazing discovery in its own right, but it had its limitations. There was only one disease that inoculation with cowpox could protect you against, and that was smallpox. But that idea that you could provide infection with something that's not pathogenic, which then creates memory to something that is pathogenic, was getting ready to be much more broadly applied idea. And we have Louis Pasteur and an accident in his lab to thank for the first immunization, which actually was against avian cholera. So avian cholera is a highly virulent chicken disease, and Pasteur was studying it, and he was studying the etiologic agent Pasteurella multocida, which is pictured here on blood agar and chocolate agar. Blood agar you're familiar with. Chocolate agar is actually partially hemolyzed blood, and so some things that don't grow as well on blood agar will grow a little bit better there. But this disease causes massive die-offs of birds. It infects their gastrointestinal tract, the lungs. The picture you see here is multiple abscesses all over chicken lungs. So they're studying this disease. They realized that they could inoculate chickens with this particular microbe and cause disease, which you'll remember from Koch's postulates is a very important step in understanding the etiologic agent of the disease. He studied the transmission of the disease. He identified asymptomatic carriers of the disease. And in 1879, he observed purely by accident that cultures that were grown over time outside of a chicken host lost their virulence, lost the ability to cause disease. And the way that this happened apparently was that he and his assistant were getting ready to go on vacation. I think it was for Easter. And he had asked his assistant to inject the chickens with a fresh culture of the bacterium before he went away on the holiday, and the assistant forgot. And when he returned a month later, he realized he had forgotten, so he took those cultures that were a month old and inoculated the chickens, and unexpectedly, the chickens only showed very mild signs of the disease and survived. Thankfully, this assistant wasn't so horrified at the thought of telling Pasteur he'd done something wrong that he kept it from him, because he told Pasteur what happened, and this really intrigued Louis Pasteur, and he started thinking, I wonder what this means. And what he did was then inject them with fresh bacteria, and the chickens did not become ill, and that was our first immunization. The attenuation of a pathogen to a non-lethal form, and then the injection of that non-lethal or non-pathogenic form in order to create immunological memory, which then reacts against the pathogen at a later time. So 1700s, 1800s, and really this period of time in the late 1800s is when vaccinology as a science really just started to boom. So the vaccinology boom occurred from about 1875 to 1900, and it includes Work by Louis Pasteur discovering that ability to create attenuated bacteria, which he took from avian cholera and then applied to anthrax and rabies. It included some work by Robert Koch, who mastered pure culture technique and discovered the cholera and tuberculosis bacilli and formed his postulates, which you'll remember from chapter 14. It included work by von Behring, who discovered diphtheria and tetanus toxoids, so creating non-pathogenic toxins which could be inoculated into people to protect them from toxin pathology. Paul Ehrlich, who developed chemotherapy, not for cancer, but the use of chemical agents against microorganisms and antibody quantification. It included work in 1931 after that sort of boom where Goodpasture and his group learned how to grow viruses in embryonated eggs, which opened up the field for people to be able to attenuate viruses. And that work was carried on by Enders, who grew the polio virus in embryonic tissue and paved the way for vaccination of polio, and then used what he had learned in 1963 to license a measles vaccine. And it's thought that the work of these men and many other scientists have prevented over 103 million childhood infections, which is excellent news. If we could just take a minute to talk about the reduced morbidity and mortality of vaccine preventable diseases, especially in diseases of childhood, which of course are called diseases of childhood because everyone contracted them as a child. They either died as a result of that disease or they had lifelong immunological memory and did not contract that disease in adulthood. So diphtheria, measles, mumps, pertussis, which is whooping cough, polio, rubella, smallpox, tetanus, these are all diseases that had a high death rate prior to their vaccination. And for each of these diseases, you can see in the far right-hand column that the deaths due to these illnesses have been reduced by 99 to 100% for each of them, which is really exciting because not only were these terrible diseases to survive, they were oftentimes diseases that killed people. For instance, measles, about 1 in 1,000 people contracted brain damage, and 1 in 500 died of that disease. So even those who survived could have had brain damage as a result of the infection. So the fact that these vaccines are available and can help to prevent some of these childhood diseases is really an amazing thing. One disease that's had such a successful campaign of vaccination that it's nearly eradicated is poliovirus, which causes gastrointestinal tract infection and in some people moves from the gastrointestinal tract into the neurons. And when it gets into the neurons, it can damage them and cause paralysis, which is lifelong. It can also cause paralysis of breathing muscles. So you might remember pictures of people in iron lungs as they were being treated for polio because they couldn't breathe. And in 1988, there were very few countries that had eliminated polio. As you can see from the diagram on the left-hand side, nearly the entire globe is red, which are countries that have never eliminated polio. By 2016, there are only 3 countries in the world that have never eliminated polio. And if you look at the numbers since then, it's actually even more exciting than that. In Nigeria, there has not been a reported case of polio since 2016. So Nigeria is getting toward eradication as well. And pretty soon, hopefully it'll just be Afghanistan and Pakistan which have polio, and then hopefully very soon after that it will be nowhere in the world and we can celebrate the worldwide eradication of a second infectious disease. Now, having said all that, I think it's only fair to say that vaccines are not a perfect solution to infection. They do come with risks, and the poliovirus campaign is one of the places where you can really see that risk playing itself out in something called vaccine-derived poliovirus infection. And to understand what that is, we need to talk just a little bit about the vaccines that are used to combat poliovirus infection. So there are two vaccines. One was licensed in 1955, and it is an oral attenuated virus. So it's been attenuated so that it doesn't cause polio, but it's still alive. And the benefit to that is that this virus, because it's alive, will elicit an immune response from both the humoral branch of the immune system and the cell-mediated branch of the immune system, because it will infect the epithelium of the gastrointestinal tract, but because it's been attenuated, it can't move into the neurons and cause neurological disease. The injected inactivated vaccine was developed in 1961, and as its name suggests, it's inactivated, but because it's inactivated, it can only mount a humoral response. So the oral vaccine provides a greater immune response, and so it's really useful where polio is still endemic. So where polio is still endemic, the oral vaccine is used. So in places like Afghanistan, Pakistan, Nigeria, in places like the United States where polio has been eradicated, we've switched from the oral vaccine to the injected vaccine. Now, the problem with the attenuated vaccine is that some of them have reverted to a more wild type form and they actually cause polio. So vaccine-derived poliovirus infections happen as a result of someone having been given the vaccine, and the rate of infection is about 3 per million poliovirus vaccinations as opposed to a rate of 5,000 per million infections if you were to contract polio. So this is not without its dangers, but obviously 3 per million as opposed to 5,000 per million means that you have a much lower odds of getting sick from the poliovirus vaccine than from the poliovirus itself. The current childhood immunization recommendations in the United States involve 12 immunizations for diseases such as tetanus, diphtheria, pertussis, Streptococcus pneumoniae pneumonia, Haemophilus influenza B meningitis, hepatitis A, human papillomavirus, which can cause cervical and pharyngeal cancer, influenza A, hepatitis B, the measles, mumps, rubella immunization, chickenpox, and rotavirus. Some of these, as you can see here, provide immunity to toxins. For instance, the tetanus and diphtheria immunization is not to the bacteria themselves, but to the toxin that they produce. And combined with those tetanus and diphtheria toxoids are acellular fragments of Bordetella pertussis, which causes whooping cough. So these are not capable of causing infection because they actually are not attenuated bacteria. They are portions of the bacterium. Streptococcus pneumoniae pneumonia is a polysaccharide conjugate, as is H. influenza type B meningitis. Hepatitis A is an inactivated virus. Papillomavirus is just antigenic fragments of the virus. Influenza A has really interesting injection, is an inactivated virus. The nasal mist that you inhale is actually an attenuated virus, which can grow in the nose but can't grow at body temperature. Measles, mumps, and rubella are attenuated viruses. Chickenpox is an attenuated varicella zoster virus, and rotavirus is an attenuated rotavirus. And it's estimated that if you were to get just 7 of these 12 immunizations, that will prevent 33,000 deaths and 14 million infections at a cost savings of about $10 billion in direct costs and $33 billion in indirect costs. And it's thought that this immunization schedule has prevented 20 million deaths due to these childhood diseases worldwide from 2010 to 2015. I mentioned in the last slide several different kinds of vaccines, which probably sounded a little bit like gibberish to you, but just to recap what those are, attenuated vaccines are live viruses or bacteria which have been altered to remain alive but incapable of causing infection. So that would be like the oral polio vaccine, the measles, mumps, rubella vaccines, those are capable of growing, and the good news about that is that they're able to get into the cells and cause both a humoral and cell-mediated immune response to them. The second kind of immunization that we talked about are inactivated vaccines. So these are either dead bacteria, inactivated viruses, or maybe even just immunogenic portions of the microorganism or virus pieces, protein pieces, or inactivated toxins, or polysaccharides or oligosaccharides from the outer membrane of bacteria that are immunogenic but are non-pathogenic. And then we have these sort of theoretical and cutting edge vaccines which don't exist for humans yet, but they involve injecting people with simply the genetic material from the pathogen, right? So just a couple of genes so that it will kind of mimic the infectious process where the virus would bring its DNA into the cell and then make the protein and then use the protein to make the virus. Well, if you only allow the cell to see one single viral protein, it's never going to produce viruses, but it will produce those proteins and then transport them out to the surface of the cell in their MHC class 1 molecules. And you'll remember from chapter 17 that cytotoxic T cells come over, interact with that major histocompatibility 1 molecule, recognize that foreign protein and mount an immune response to it, which means that should the entire virus ever find its way into the body, that immunological memory will be able to come on board so quickly that the inoculation with the pathogen will produce an inapparent infection. Before we move on to another topic, I would like to take a moment to talk about a really interesting phenomenon called immunological amnesia that happens as a result of measles infection. So prior to immunization for measles, about 4 million people died per year from measles infection. But what was really interesting once the measles vaccine was licensed was that non-measles infectious disease mortality went down drastically. And so people started looking into why that might have been. And one piece of evidence was found in the work of a researcher named von Perket, who in 1908 noted a lack of the tuberculin-Perket reaction, which today we know as the PPD, where you have tuberculin protein injected under the skin in order to see if you've been exposed to tuberculosis. Most of you are currently or will be in the healthcare profession, and you will have that test done repeatedly to make sure that you are not an asymptomatic carrier of tuberculosis. And what he noted was that people who had contracted measles lost that immunological memory to tuberculin. And what they realized was measles, because it's capable of infecting immune system cells, causes what's called immunological amnesia. And so measles infection became implicated in not just the deaths due to measles, but implicated in half of all childhood deaths due to infectious diseases prior to the measles immunization. And the measles immunization has reduced overall childhood mortality due to infectious diseases anywhere from 50 to 90%. And that all relies on the fact that when the measles virus infects immune cells and those cells die, any memory those cells contained is lost to the body. And that was found using a really interesting experiment that incorporated the green fluorescent protein that we used in our transformation experiment. So they had a measles virus that they had bioengineered to have this green fluorescent protein expressed in any cell that that measles virus grew in. As you can see from the pictures here, mesenteric lymph, the gut-associated lymphoid tissue or GALT, and the Peyer's patches all lit up bright green, meaning that the measles virus was attacking those immune cells and they were dying, and the immunological memory was dying with them. Long-term measles-induced immunological amnesia results initially because the number of lymphocytes are drastically reduced as measles affects those cells, infects those cells, and kills those cells. But it also reduces the number of innate immune cells, and it preferentially attacks the central memory and effector memory cells for both helper T cells and cytotoxic T cells. And it's thought that that immunomodulation, the loss of immunological memory, lasts about 27 months. So the take-home message is that measles, which sometimes people refer to as sort of a benign childhood disease, is in fact not benign at all. And the ability to stop measles infection means that you're preserving the immunological memory that results either from infection with other infectious agents or from immunization with other infectious agents.
Where a young woman contracted varicella zoster virus, which is commonly known as chickenpox. So this is a gal who's 15 years old, perfectly healthy, never been immunized for chickenpox. And on March 12th of 2009, she was admitted to the ER with a 3-day history of rash, a 1-day history of shortness of breath, and what would be considered a mid-grade fever. And her diagnosis at that point is septic shock, so they admit her. And during her hospital stay, her treatments include acyclovir, which is an antiviral medication, and various different antibiotics, as well as mechanical ventilation, because upon admission into the hospital, her shortness of breath quickly escalates to an inability to breathe on her own. So at this point, her diagnosis is expanded to include pneumonia and acute respiratory distress, multi-organ dysfunction, pancytopenia, which basically means that you have red blood cells, white blood cells, and platelets. So pan meaning all of the cells of your from your bone marrow are low, right? Because penia means small, cyto means cell, pan means all. So all of these blood cells are at low levels. And this is typically caused by ineffective T cell activation, because what happens is the T cell actually activate and cause macrophages to engulf all of these blood cells, the red blood cells, the white blood cells, and the platelets. So it's very serious. And in addition to those symptoms that are directly caused by this varicella zoster virus, she also ends up with 3 nosocomial infections, a lower respiratory tract infection of Enterobacter cloacae, which probably was a result of that mechanical ventilation. She ends up with a urinary tract infection with Pseudomonas aeruginosa, which is a result of her being catheterized. And she ends up with septic infection of Stenotrophomonas maltophilia. And on day 21 of her infection, this young, otherwise healthy gal dies of what began as a chickenpox infection. So all of that just to say that even in today's day and age, with modern medicine, quick response to infection, there are still deaths that can be attributed to these diseases that are preventable with immunization.
Well, if one case study is fun, then 2 is really fun, right? So let's take a look at a 2017 case study for Clostridium tetani, where we had a 6-year-old male who had a forehead laceration. He bumped his head, his parents irrigated the wound, cleaned it, and stitched it. And he seemed fine, but about 6 days later, he started having muscle spasms and jaw clenching, and his back and neck would arch, sort of as is pictured here, and he started having breathing difficulty to the point where he was airlifted to a hospital. And when he was brought into the emergency department, he seemed alert, but he was unable to open his mouth to drink. He was in respiratory distress to such an extent that he had to be sedated, intubated, and put on mechanical ventilation, and he was diagnosed with tetanus poisoning. And the treatment for that was to give him the tetanus immunoglobulin, so an antibody against this tetanus toxin. They also administered the DTaP shot, which includes the tetanus toxoid. They had to also put him in a dark room in the PICU with earplugs because he was so agitated by sensory stimuli. They debrided the wound, they gave him IV antibiotics to kill the bacteria, and they had to give him a neuromuscular spasm blockade in order to allow his muscles to relax, attaches to the ends of neurons and stops them from relaxing, and so it causes a spastic paralysis. He was on a ventilator for about 7.5 weeks. So on day 44 of infection, the ventilator was discontinued. On day 47, he was transferred from the PICU to an intermediate care unit. On day 57, he was transferred to a rehabilitation center for 17 days. And finally, on day 87, he resumed normal activities. So all in all, this forehead laceration through which he contracted Clostridium tetani, which is common in the soil and on metals and things like that. I think he hit his head on a tractor or something like that. At any rate, required 57 days of acute care, 47 of which were in a pediatric ICU, and the hospital bill alone was $811,000. As we mentioned in the diversity unit, there are multiple different ways of identifying unknown microbes. One of them, which we use quite commonly in lab, is the microbiological route, which includes differential staining and physiological testing, which gives you a number of the characteristics of an unknown cell and allows you to compare those to a known cell. What we're talking about in this unit is called the immunological route, which is shown in blue, which historically began with a blood sample, and that blood sample was used to search for antibodies against a suspected pathogen. And we're going to be discussing different ways that immunological assays have gotten better and better and better at finding antibodies, and it turns out antigens, in patient samples in order to have a highly reliable measurement of whether or not a person has contracted a particular etiologic agent. And as we move through those, we're going to be looking at the fact that as those tests have become more sophisticated, they've been able to correctly identify positive results when very little level of positive results are available. But before we get to those tests, I want to talk a little bit about the immunological vocabulary. Two different kinds of immunological identification assays are performed in labs routinely. Both of them rely on looking at the interaction of antigen and antibody. Some look for antigen in the patient, and those are called direct immunological assays because you are directly looking for the antigen, and others, which try to detect antibody in the patient, are considered indirect because you're using the presence of the antibody and assuming that the presence of the antibody indicates the presence of the pathogen as antigen. So when you do this, it's very important that you differentiate whether you're talking about having IgG, which are indicators of immunological memory to past infection, or IgM, which are evidence of an acute infection which is being fought in real time in the patient. Either way, these two types of immunological assays have statistical descriptors which give us an idea of how well they work in order to identify positive tests and also negative tests. So two statistics that you'll sometimes hear are sensitivity and specificity. Sensitivity describes the probability of a false negative. So sensitive, the more sensitive a test is, the more you can trust the negative. Sensitive, when negative, rules something out. So if you have a 100% sensitive test, you can absolutely trust the negative result. There is nowhere in there a false negative, a person who actually has the disease, but that you don't think they do. So sensitive tests allow you to trust the negative. Specific tests describe the probability of a false positive. So the more specific a test is, the more you can trust the positive results. So specific tests, very specific tests, when positive, rule in a disease. So whereas a 100% sensitive test will tell you that that person, the no actually means no, a 100% specific test will tell you that a positive result is 100% positive. Two other related statistical terms are positive percent agreement, and basically that again says if the test is positive, the person is positive for that infectious agent, and negative percent agreement, which says if the test is negative, they are negative for it. And there are some differences in the way sensitivity and negative percent agreement and specificity and positive percent agreement are determined, but isn't a statistics class, so we're not going to get into that. What we're going to do instead is look at a couple of different ways that tests, immunological tests, have become more sensitive and more specific over time. And really, one of the things that we're going to look at that has been a huge advancement since immunological assays were identified in the early 1900s is being able to see a positive result, coming up with a positive result that is visible no matter how few antibodies and antigens are interacting. So as we look at these, you're going to see increasing specificity and a lack of false negatives because the positive, when it is there, is always visible. Before we do that, though, we have to talk about what to do when you're trying to detect antigen in a patient and you need to bring with you antibody to it. It's very easy to imagine a scenario where we want to detect antibodies in a patient. We grow the bacterium or the virus in the lab, we add the patient blood serum to it, and we look for that agglutination reaction, which we learned about in chapter 17 as a result of antigen-antibody interaction. But if you need to detect antigen in a person, then you need to bring with you the antibodies to that antigen. And the way that you do that is by producing something called a monoclonal antibody, which is the topic of our next slide.
Monoclonal antibody preparations contain only one type of antibody, derived from a single cloned B cell. Consequently, they are highly specific for a single epitope and have applications, for example, in diagnostic microbiology and cancer therapy. The first step in producing a monoclonal antibody is to inject an animal with an antigen containing the specific epitope of interest. Each B cell produces a single type of antibody. B cells are isolated from the spleen and then mixed with myeloma cells, a type of cancer cell that grows continuously. Addition of polyethylene glycol causes the two types of cells to fuse together to form cells called hybridomas. The mixture of B cells, myeloma cells, and hybridomas are cultured under conditions which permit growth of only the hybridoma cells. Each hybridoma cell will produce a single type of antibody against a single epitope. The single hybridoma cells are then separated into individual wells of a microtiter plate and tested for ability to produce the desired antibody. The hybridoma cells producing the desired monoclonal antibody are then cultured. Monoclonal antibodies are isolated and purified.
The technology for producing monoclonal antibodies was developed, there still were immunological assays, but they relied on the fact that you were doing an indirect test where you had a patient sample from whom you were wondering whether there was an immune response, and you were looking for that by using a lab bacterium or other microbe. The first recorded agglutination reactions that we have happened at about the turn of the 20th century, and they were done by two scientists who noted that when you combined blood serum from people who had contracted typhoid fever with Salmonella typhi bacteria, the blood would clump, and they termed that clumping reaction, agglutination, that was sort of finalized and sophisticated by a French scientist, Woodall, who found not only could you determine the status of a person who was infected and showing symptoms of typhoid fever, you could actually use that same agglutination reaction to diagnose asymptomatic typhoid carriers. So in either case, Salmonella typhi bacteria were mixed with a blood sample from a patient, and agglutination or clumping was a positive reaction, and a lack of agglutination was determined to be a negative reaction. This, incidentally, was also the basis for ABO blood typing. So agglutination relies on the naked eye being able to see a clumping reaction between antigen and antibody. So the question is then today, how do we make the positive reaction more visible in an agglutination? And the answer to that is latex beads. Number one, the latex bead is larger than an individual antigen or antibody, and the latex bead is often colored. So on the left-hand side of your screen, you see latex beads to which antigen have been attached, and those antigens are mixed with a blood serum from a patient which contains IgM antibodies, and that clumping is made more visible and more sensitive because the latex bead is more visible than the antigen-antibody interaction itself. So this would be an example of a positive indirect agglutination reaction because you're looking for antibody in the patient. On the other side, we have latex beads to which antibody have been bound. So again, a colored large latex bead is used, but this time it has antibody bound to it, which are looking to bind with antigen from the patient's sample. So whereas we had a positive indirect test on the left-hand side, we have a positive direct test on the right-hand side. And one common serious infection that is diagnosed using these latex agglutination assays, Cryptococcus neoformans, which is the causative agent of pneumonia and meningitis in immunocompromised individuals. Another more common test that's done in doctors' offices is the latex test for Staphylococcus aureus in different wounds. As you can see from this diagram, the positive on the left-hand side shows obvious clumping, which is made much more visible because of the red latex beads, as opposed to the negative test on the right-hand side, which just sort of has a pink haze. So clumping on the left, an example of a positive result, no clumping on the right as an example of a negative result.
So if we look first at the left-hand side of the slide, you see two different situations. At the top, you have group A streptococci from a patient's throat, which would be collected by swabbing the back of a pharynx, just like you did to yourselves during your strep lab. You would actually smear that onto a microscope slide, and if you wanted to, you could look at it under the microscope right there. You could even gram stain it, and you would find that you'd have gram-positive cocci, but you would not know what gram-positive cocci you were looking at, and you would not know whether or not that was pathogenic and was likely causing the patient's symptoms. So instead of gram staining or simple staining that patient sample, you instead stain it with a fluorescent dye labeled antibody that will bind to the streptococci because of the variable region of the antibody, but will fluoresce under the microscope because the antibodies are fluorescent. So you stain it with these antibodies, you wash it off so that all antibodies that are not bound to antigen wash away, and then you look at it using a special kind of microscope, and you can see there right at the end of that left-hand picture those glowing green streptococci. So again, we can see their morphology and arrangement, but we can also tell that they are exactly the microorganism we were wondering about because the antibody is bound to them, and that antibody interaction with antigen is so sensitive that it would not occur if that were not the actual antigen for that antibody. So this, because we use a microscope and the fluorescence increases the visibility of the positive result, we get a much more sensitive result. We have a much lower likelihood of a false negative. Now that right there, because you're asking for antigen from the patient sample, you are talking about a direct test using fluorescent antibodies. We can also do an indirect test using fluorescent antibodies. So in that case, we're looking for antibody from patient. So in order to have that antigen-antibody interaction, we provide the antigen. So here you have an example of Treponema pallidum, which is the causative agent of syphilis, from laboratory stock, right? Not from the patient, because we're looking for antibody from the patient. So you put T. pallidum on the microscope slide, you wash the slide with patient serum, and if the test is positive, then you have the binding of the antibody to the T. pallidum, right? If the patient did not have antibody to T. pallidum, then all of the antibody would wash off, right? So at this point, you have a positive result, but you have a positive result that is invisible. You can't see those antibodies. So you have to move to what we call in these immunological assays an indicator step. And an indicator step is one which makes the positive visible. So in all of the cases subsequent to this, including this, the indicator step is the one that makes the positive visible. So the indicator step in the fluorescent antibody test is to add fluorescent antibodies, and it's very important that you notice that those antibodies do not bind T. pallidum, they bind the constant region of the antibody. So they bind to the patient antibody, and if there was no patient antibody there, then they would not bind, they would get washed off, and you would see nothing on that microscope slide, right? So they do not bind antigen, they bind the antibody that the patient provided. And there we have an example of a positive indirect fluorescent antibody test. Now, these can also be used in very specific cases where you actually use flow cytometry, which we talked about in chapter 6, as a way of counting bacteria. You can take those fluorescently labeled bacteria, and rather than looking at them on a microscope, you can throw them through a flow cytometer, which will shoot a laser across the drip that's coming out, and every time it catches a cell that fluoresces, it counts it, and that can give you a highly specific, right? So you're not going to get a false negative. You have a highly specific way of determining whether or not a person has either antigen or antibody to a given microorganism. So the fluorescent antibody technique is better, I could say, than the agglutination reaction because you don't have to have as much antigen and antibody interaction in order to see the positive result. And now we're going to see a case where we get even more sensitive than the fluorescent antibody technique.
Here we see another example of the positive reaction being made more visible. In this case, the positive reaction is made more visible not by the use of fluorescence, but by the use of red blood cells, which, because they're large and colored, are usually very easy to see with a naked eye. In order to understand this particular immunological assay, complement fixation, it's necessary to remember back to both chapters 16 and 17, where we learned about complement and what complement does as part of the innate defenses of the body. Complement, you'll remember, is one of the innate defenses that can be activated in 3 different ways, and upon activation can do 3 different things. You might remember those 3 things are opsonization, the enhancement of phagocytosis, the initiation of inflammation, and then something called cytolysis, which is pictured at the bottom left-hand corner of this slide, where complement proteins, upon activation, will punch holes in the wall of a pathogen and cause that pathogen to lyse. Now, one of the ways that cytolysis can be initiated is by antibodies, which have first bound to antigenic structures on the surface of the cell. And that's what's pictured down here in the bottom left-hand side of the slide. You see the two green antibodies bound to the surface of the bacterial cell, the complement proteins recognizing the constant region of the antibodies, and then becoming activated and lysing the cell. So we're going to have to keep that in mind as we look at this complement fixation, which is used today in order to test for antibodies to the organism Borrelia burgdorferi, which is the causative agent of Lyme disease. So again, as you're looking for antibodies from the patient, then you'd be doing an indirect complement fixation test. The test here is shown with a positive result on the left-hand side and a negative result on the right-hand side. You'll see up at the top that what you bring from the lab is the antigen as well as complement, and those are added to blood serum from the patient. Now, when the blood serum from the patient contains antibodies, you'll see that that very similar setup of antibodies bound to antigen and complement bound to antibodies sets itself up there very nicely. And of course, we know that when that happens, the complement becomes activated and it lyses that antigen provided by the lab. So at that point, we have an invisible but positive reaction, which we can compare to the right-hand slide, where we have the antigen and complement, which are added to patient blood serum, which does not contain antibody. And so there is no complement fixation, and we have on the right-hand side of the diagram an invisible but negative reaction. So here comes what we call the indicator stage. The indicator stage is where something like the fluorescent antibody in the previous example is added in order to make the positive and negative visibly distinct from one another. And in this case, we're going to do something kind of strange, and we're going to add sheep red blood cells and antibody to sheep red blood cells. Now, without even looking at the bottom of the slide, you should be able to picture that the antibodies to sheep red blood cells will bind to the sheep red blood cell as soon as they come into contact with one another, regardless of what else is going on inside this complement fixation reaction. So on the left-hand side, you see just that. You see these antibodies bound to the red blood cell antigen. Notice, though, that the complement does not bind to the constant region at the back end of the antibodies because complement is already tied up in that antigen-antibody reaction with antibodies from the patient. And so in this case, a lack of hemolysis is indicative of a positive result. And you can see in the lower right-hand side of the slide, the top of a microtiter well, you're looking at a top view and you see what kind of looks like a red button, and that red button is indicative of a positive result because the red blood cells have not been hemolyzed and they clump together at the bottom of that microtiter well. On the other side of the diagram, we have the negative test, and when you add the sheep red blood cell and the antibody to sheep red blood cell together in the negative test, the complement, which has not been combined with any antibodies previously, will very quickly attach to the constant end of these new antibodies and lyse the sheep red blood cells, resulting in this hazy red color, which you can see both in the diagram and also on this side of the slide over here, right? So here's a negative, here's a positive reaction. So the button shows us the positive reaction, and that hazy pink color shows us the negative reaction. Our next immunological assay enhances visibility by combining an enzyme-linked antibody which recognizes a positive result and produces a visible color change. That happens because the enzyme is capable of converting colorless substrate to a colored product. Because an enzyme converts substrate without being altered itself, it continues to convert substrate over and over again, making an ever more visible positive result. Just like the other assays we've been looking at, the ELISA comes in both direct and indirect forms. On the left-hand side of the screen, you see a direct ELISA, called that, of course, because it's looking for patient antigen. In a direct ELISA, an antibody specific to a pathogen is adsorbed or adhered to the bottom of something called a plastic microtiter well. And when the patient sample is added, the antigen from the patient will be bound and trapped by that monoclonal antibody. A second monoclonal antibody to which that enzyme has been linked or attached is added, and that recognizes and binds to the positive patient sample because it also recognizes the pathogen as antigen. A colorless substrate is added in step 4, which is converted by the enzyme to a colored product, and you get a visible result that becomes more visible as the enzyme continues to convert the substrate. On the right-hand side of your screen, you have an indirect ELISA, which begins with a laboratory-bound antigen bound to that microtiter well, because this test is looking for patient antibody. If the patient sample contains antibody, it will bind the lab antigen, and then a second lab antibody, this one recognizing the constant region of the patient antibody, is added, confirming that positive result. Now, in both the direct and indirect ELISAs, you'll notice that the enzyme-linked antibody is recognizing and binding to what the patient sample has brought in order to prevent false positives. So again, once the enzyme-linked antibody has done its job of recognizing the positive result in step 3, the enzyme will do its job in step 4 of converting the colorless substrate into a colored product, making every positive test visible and allowing this test to be incredibly sensitive because every positive test is visible and there are virtually no false negatives. In order to see this process happen dynamically, which I think makes it a little easier to understand, the next slide I'm going to show you is a video of this process occurring.
The enzyme-linked immunosorbent assay, or ELISA, is a commonly used format for serologic testing. The purpose of this animation is to explain how this test works. ELISA serologies are usually done in multi-well microtiter plates so that dilutions of serum can be easily prepared and tested. To better understand how this assay is done, let's take a closer look at what happens in one of the wells of this assay plate. To perform the assay, the wells of the plate are coated with the antigen of interest. For commercial tests, this would be done by the manufacturer of the assay. To begin testing, the wells are filled with dilutions of the patient's serum. If antibodies against the antigen are present, they will bind to the antigen fixed to the bottom of the wells. But only antigen-specific antibodies will bind to the wells. The wells are then washed out to remove all unbound antibodies. Next, a solution of an animal antibody against human antibodies is added. This second antibody is covalently conjugated to an enzyme. The wells are washed again, this time to remove any unbound enzyme-conjugated antibody. Finally, a solution of a colorogenic enzyme substrate is added. The interaction of the substrate with the enzyme on the second antibody generates visible color.
One final and really cool immunological assay is something called a lateral flow assay, which enhances visibility by conjugating antibodies to a pigmented molecule. And this one's used diagnostically in order to determine whether or not a person who has pharyngitis has streptococcal strep throat caused by Streptococcus pyogenes. So the problem is when you go to the doctor and you have a sore throat, about 70 to 85% of those pharyngitis cases are viral, for which no antibiotics should be prescribed. And even of the 15 to 30% which are bacterial, most of those are going to be self-limiting, in which case, again, you don't need to take antibiotics. But Streptococcus pyogenes is also a causative agent of pharyngitis, and it has the long-term sequelae associated with infection that include rheumatic fever and kidney disease. So if you have S. pyogenes causing pharyngitis, you want to make sure that you take antibiotics. But the problem is that clinical criteria alone cannot diagnose those group A beta hemolytic strep pharyngitises from other pharyngitis. So what your clinician can do is take a swab from the back of your throat, which will contain antigen, place it in the sample pad of one of these lateral flow assays, and add some of these assay liquids to it, and the sample will start to move through the lateral flow assay, and if the antigen is present, then it will grab and attach these conjugate labeled antibodies to it, and then as it moves through the lateral flow assay, those antigens which have been grabbed by these antibodies will be bound by a second antibody, and that will concentrate all of those colored conjugates in a line which can be read as a positive test. So at the end of this test, you end up with two red lines if you have a positive test. The line adjacent to the C is the control line, which you'll notice is past the test line, which in the case of a positive test is also colored, and it really doesn't matter whether that color in the test line is dark or light. A colored line means that antigen was able to be sandwiched between those two antibodies, much like the antibody sandwich of the ELISA, which was just described to you. In a negative test, because there are no antigen present, nothing will bind to the test antibody test line, and all of the conjugate will move to the control line, where it will color only the control line red, and you will have a negative test. So again, we're seeing increased visibility, helping us to rule out false negatives.